In this video, we will discuss about this effect of SARS-CoV-2 virus-like particle with the N-glycan mutation on the spike protein. So these are the four plasmid that are used for this experiment, that whose results are shown in this figure. So each plasmid has a different reporter. That is this uh, first plasmid. This is the envelope plasmid, has this flag reporter. This spike plasmid has this EGFP or flag reporter, and this third plasmid, that is membrane plasmid, has this HA tag. The fourth plasmid, that is nucleoplasmid, has this C mice tag. So first they did the confocal macroscopic analysis when they transfect these cells in the HEC293 cells, and this is the individual figure that shows that uh, this nu nucleocapsid, spike, membrane, and envelope plasmid, all of four of them lies in the perinucleus region. This perinucleus region is the region that surrounds near the nucleus of the cells, as we can see in these figures. And this is the merged figure where all the four plasmids can be seen, and we can see that they are in the perinucleus region of the cell. So this figure shows that all of these structural proteins are formed and formed near the perinucleus region. Next, on the thing which is present near the perinucleus region, or the organelle that is present in the perinucleus region, could be endoplasmic reticulum, could not be. So next they did the flow cytometry analysis of each of these spike protein transfected in the cells, and whether they did it at two different strategies. First one is the live cells, and second one is fixed and permeabilized cells, to identify whether these structural proteins are finally secreted on the cell surface or remain within the cells. So when they find out that this membrane protein and this spike protein, membrane protein and the spike protein are expressed on the cell surface more, and this also membrane protein, spike protein, and this is also the membrane protein, and this is the combined all the first four is the individual transfection of the proteins, and this fourth is the common transfection. When common means all the four plasmids are transfected in the same cell system. So in this they have find out that spike protein and the membrane protein, they are the two proteins that are expressed on the cell surface. And when they did the same things during the fixed and permeabilized cells, they have find out that all the proteins are expressed. This means that uh, this envelope protein and the nucleocapsid proteins are expressed more on the intracellular part of the cells. So next they did, so this is the analysis of flow cytometer. Thereafter they do the Western blot of all the membrane proteins, with the different mutation in the spike, different mutation in the uh, SARS-CoV-2 spike protein. And this time they find out that this expression of N61Q mutation and N801Q mutation is reduced compared to the uh, previous study, in which uh, they have not introduced all the four spike proteins together. So what they have seen is that all the four proteins are expressed within the cells in the different condition. whereas the spike proteins that are expressed more are this N603Q and N603Q and 657Q, these mutations. And apart from this, the all the four structural proteins are expressed within the this HEC293 cells. Later on, what they did is they change the plasmid. They change the this plasmid, or we can say spike plasmid, or modify the spike plasmid with luciferase 2 and PS9, luciferase 2 reporter, and this with PS9 uh, construct. And what they have find out this time is that the fluorescence of this N60, or we can say luminescence of this N603Q is increased and N616Q is increased, whereas the N61Q and N657Q is reduced, and in all the other case also it is reduced. The expression of this spike protein is reduced. So this shows that these mutations could be quite important, that is N603Q and N616Q for the viral infection, and these mutations such as N61Q and N657Q and 801Q are also important in terms of reducing the viral infectivity. So later on, what they did is they again did the analysis of this luciferase, this modified spike construct, in a way that they treat the spike construct with different different enzymes such as this endovH or PNGase H. EndovH usually cleaves high mannose structure, and whereas PNGase cleaves all the glycan structure, to check whether there is a role of spike glycans on the spike protein incorporation. And when they did it with the control, they do not see any uh, signal for non-spike injection. And when they treat it with the spike proteins that are treated with EndoH, they find out that there is there is not a slight broadening of the signal. This indicates that some of the high mannose structure could play a role, and this leads to the to the broadening of the band, because now the high mannose structure is not there. Because of that, the difference in the spike proteins could have increased. That leads to the broadening of this signal, we can say NTS2 signal. Next, when they treat the cells with PNGase F, they can see the wide broadening of the signal, and also the faint of the signals indicate that the production spike protein production might have reduced. Spike protein production might have reduced because of the cleavage of glycans from the spike proteins, and that will leads to the broad these signals to spread across the broad range, because the removal of the glycans, that will leads to change in the structure, molecular weight, and thereby they will move faster in the bands compared to those spike proteins in which the glycans still exist. Later on, they did this thing, analysis on two kinds of cells. First one is the 293 H2 cells with H2 cells, and next second one is the Kalu3 cells. And there also they see the similar results that with endo H there is a slight reduction in the signal luminescence, whereas with PNGs there is a maximum reduction in the luminescence. And same thing is happened when they did the experiment with Kalu3 cells. This shows that the SARS-CoV-2 infection or SARS-CoV-2 entry within the cells is also dependent on the glycans that are present on the spike proteins of the SARS-CoV-2. So if you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.